Thank you very much, and thank you very much for having me here. And I'm really truly grateful for Human Frontier Science program. It might be fair to say that my entire career is almost like sort of back, and I'm really thankful because like a lot of the changes I made was supported and encouraged by the program. So let me introduce a little bit of that. So I started my PhD in tRNA modification enzymes. So I was very interested and fascinated by this molecule of tRNA. I still believe that's really the core of the life that connects the nucleic acid information and the protein sequence. And at that time, I was very curious also, why does life and cells use this very core fundamental molecule to produce local proteins at neurons to sort of gain functionality in synapse and the brain function. So I wanted to change my field to neuroscience, and there I first gained the access to the HFSP long-term fellowship that really supported me to convert myself to start neuroscience in the US, and that was really fascinating. And there I could learn a lot about how the neurons form its shape, how they connect, and all those things, fascinatingly coordinated with the genes and proteins that are locally and globally produced. And that really made me interested in how neurons connect and how they form the circuit, and then they entirely get the function at the end. But that's really fascinating. At the same time, I was quite struggling the idea of why we cannot make those culture neurons functional in the dish, because like day to day I culture those neurons in the lab, but I really couldn't get the hang of the function of it. So I came back to Japan, and then I started this engineering of those neurons as a tissue. And then that led to this new award of the HFSP together with the French group. So I'm going to have 2 more slides to talk about each of them. So what I'm working on is to build circuitry with the brain organoids. So organoids are the artificial tissues. They can grow from the stem cells, including iPS cells, and you can make them, and that forms intricate layers with the many different cell types that are normally seen in the brain. But it has so many limitations. It doesn't have any macroscopic circuitry, for example. So that really is a limitation. So we thought we want to connect organoids, and as an engineering ways to connect them. And then that really, we believe, is the key to unlock their function. So we see that the activity changes quite a lot if we give the structure by connecting them. Also, we can model diseases such as like a loss of corpus callosum between the left and right hemispheres with this model. And we are starting to see the ability for the tissues to gain functionality if we manage to make certain structures and also give certain sets of stimulation for long term. And we started to use this to be able to connect with the more electrical system so that we can have a very, very fast feedback loop that kind of give them a virtual body in the virtual world. So those things really we started to working on in the lab. And that it's great for us that we kind of see the light of perhaps we might be able to start giving the cultured neurons function, and maybe that makes us understand a little bit more of the brain. But it also gives us like struggle on how we really gonna understand the physiological problems, how we really understand deeper, and we are overwhelmed with the data we produce. And that here, that's where the HFSP comes in again. So we got this new grant, deciphering the impact of viral infections on human neurocognitive functions ex vivo. I think it's a little bit too much. I guess as you see, there are many like sort of words that we're sort of aiming for, but we're so excited that we finally be able to understand how the synapse forms and how they connect and how they function through the help of a viral expert and an AI theory robotics expert, and we're very excited to work on this. And thank you very much.